I'm Jane Morris, and today I'm going to cover approaches to traceability, labelling and identification of GMOs with special reference to the situation in southern African countries. These issues are interlinked because unless you can trace or identify a GMO either through a paper trail or through analytical techniques, there's no point in considering labelling options. So today we'll cover, firstly, methods of identification, then why label or identify GMOs at all, what are the international approaches to labelling, what labelling thresholds are used, what are the sampling strategies, and what is the cost of labelling. In most cases where GMOs are or may be included in a consignment, for instance a shipment of grain, identification is by means of a paper trail, and testing is only carried out as an exception. This is in part because of the complexities of sampling and testing that I'll describe later. Where there is demand for strict segregation between GMOs and non-GMOs, sometimes to certify that a product is GM-free, this can be done through identity preservation. In this case, there is separation through the whole handling process from the seed to the consumer, backed up by a paper trail, which again lessens the need for additional testing. There may also be a demand for product labelling in the absence of identity preservation. In this case, products with detectable and quantifiable traces of GM ingredients determined through accredited testing methods may be subject to labelling. But we can ask the question, why should we want or need to identify or label GMOs or segregate them at all by identity preservation? Well, firstly, we have to understand that this is not a safety issue, since all GMOs on the market will already have gone through extensive safety testing before release is approved. Under United Nations guidelines, consumers do have rights to information and to choice, which could entitle them to labelling. However, this is counterbalanced by the right to affordable food, which is recognised in the Universal Declaration of Human Rights. Costs incurred in labelling need to take the balance of these rights into consideration. If labels are to be used, then they need to be clear and verifiable in order not to mislead consumers and in order that compliance with regulatory requirements can be ensured. Most African countries are signatories to the Cartagena Protocol on Biosafety, which specifies certain labelling requirements. However, these only apply to traded shipments that may include GMOs, not to consumer products. Moreover, they only apply to living organisms, not products derived from GMOs. The protocol provides options to either state that a shipment may contain GMOs or else that it does contain GMOs. Where the does contain option is used, all the GM events in the shipment must be specified, and this requires the use of an identity preservation system, as well as the ability to test the shipment to ensure compliance. It is up to individual states to determine which of these options they implement in their legislation. The African Union Model Law on Biosafety was provided as a model for African countries to use as a basis for their own GMO legislation. However, individual countries can choose what to include in their own legislation, and the model law has been criticised for being too strict. The model law implements the does contain option, requiring traceability for each trait from its country of origin to its final destination. Moreover, the model law goes beyond the requirements of the Cartagena Protocol by applying this to not only living GMOs but also processed products. The European Union also has labelling requirements that apply to both GMOs and their products. However, it falls short of applying to products from animals fed GM feed or food produced with GM enzymes such as cheese. It also does not require labelling of some non-food products such as cotton clothing. The labelling threshold is set at 0.9% according to EU legislation, but in addition, some individual countries have also introduced provisions for GM-free labelling. In South Africa, there are two sets of labelling requirements. Under the Consumer Protection Act labelling, there is mandatory labelling for GM ingredients with a 5% threshold, with the provision to use the may contain option where testing is scientifically impracticable. There's also a voluntary option to state does not contain GMOs where the GM content is less than 1%. In contrast, the Department of Health has labelling regulations that specify the need for labelling only where GM foods differ significantly from existing foodstuffs in terms of their composition, nutritional value, mode of storage, preparation or cooking, allergenicity, or for genes with human or animal origin. These two regulations contradict each other since one is voluntary and one is mandatory. Namibia is the only other country in the southern African region that so far has official labelling requirements. The regulations to the Biosafety Act specify labelling with a 0.9% threshold, which applies to both GMOs and their products. Other countries in the region haven't yet developed their labelling requirements. The requirement for labels implies the ability to test for and detect GMOs. However, this can be quite a complex matter, and before embarking on GMO testing, it's important to ask some searching questions to determine when testing is required, where it should be carried out, what is to be tested, and why is testing to be done. So, we need to determine, is the testing required to be qualitative or quantitative? What level of statistical significance is needed? Is the testing to be done on seeds, commodity shipments, consumer products, or anywhere else in the production and processing chain? Is the testing to be done on live GMOs or also on processed products? And what level of processing is involved? Is it best to detect the DNA or its protein product? And is the aim to detect any and all GM material or to distinguish between approved and unapproved events? Is the testing being done as a routine screening technique, or is it to detect suspected regulatory contraventions? And finally, where and how will the results be reported and used? All these issues need to be resolved before embarking on any testing. One of the biggest problems in GMO detection is the issue of sampling. This, in fact, creates many more uncertainties than the GMO analysis itself. Let's look at sampling of a grain shipment where there may be a heterogeneous mix of GM and non-GM grain containing a number of different GM events. In this case, the recommended procedure is to take a number of increment samples from the shipment, each consisting of 1 kilogram of grain. Half a kilogram from each increment sample is then combined to form a bulk sample, which should be at least 20 times the sample size that eventually goes to the lab for analysis. This bulk sample is then subsampled to get the laboratory sample. Depending on the original size of the shipment, a shipment of over 500 tons would require 100 increment samples to be taken and used to produce a bulk sample of 50 kilograms. Half of each increment sample is retained separately. Then, if the results of testing from the bulk sample are close to a designated threshold for labelling, it may be necessary to test individual increment samples to increase the level of statistical significance. So, just to illustrate the type of answer you might get from the lab testing, you might, for example, test 300 maize kernels and get a negative answer. This could possibly lead you to the result that the lot contains less than 1% GMOs at a 95% probability. Conversely, depending on the test you carried out, you might get a positive result from a sample size of 2,000 kernels, which could tell you that the lot contains GMOs, but at an unknown level, so it could be less than 1%. This would require further quantitative analysis to determine the actual content at any designated probability level. The example I've given here is for sampling of grain shipments, but there are different recommendations for how to sample various products along the food chain. The reference I show here is a useful and detailed report on different sampling methods. Detection of GMOs can be done at either the DNA or the protein level. Let's look first at DNA detection. DNA can be detected in fresh, frozen, or in many processed foods. However, extraction of DNA from processed foods can be difficult, and where there are refined products such as starches, sugars, or oils, it's generally not feasible to detect DNA. DNA-based detection relies on amplification of the DNA via the polymerase chain reaction, based either on commonly used regulatory sequences or on sequences specific to known events. It can be qualitative, which can provide information on simple presence of GMOs or to determine threshold levels, or quantitative, where the absolute amount of GM DNA in a sample is determined. PCR detection is relatively expensive and it needs a well-equipped lab with trained personnel. The other option is protein-based detection, but this is only applicable to foods where the protein is not degraded. So it can be used for grains and fresh foodstuffs, as well as meals and flours, but not for foods where there has been more intensive processing. It uses immunological methods to test for specific proteins of interest. The qualitative ELISA strip test will give information on thresholds, such as less than 1%, whereas a quantitative ELISA plate assay can determine the actual amount of GM protein in a sample. It's relatively cheap and simple to carry out. In reality, the majority of testing is done using PCR rather than protein testing. For PCR testing, primer selection can be based on commonly used regulatory sequences such as the cauliflower mosaic virus 35S promoter sequence and the NOS terminator sequence. Alternatively, it can be based on specific target sequences to test for particular events. However, it should be borne in mind that a negative result may mean only that inappropriate primers were used. In all cases, the results have to be interpreted at a particular level of statistical significance. Generally, qualitative screening is carried out first, and this would be followed only where necessary by quantitative screening or screening for particular events. One major issue that has arisen internationally is how to express results. Most countries have specified thresholds for labelling without specifying the units of measurement for these thresholds, and this has caused much confusion. Initially, it was assumed that the results would be expressed on a mass-to-mass or weight-to-weight basis, determined as percentage mass of GMOs in the total. However, this can lead to overestimation of the GMO percentage when there are stacked traits, since each GMO contains more than one DNA insert. Another problem is that different cultivars contain different DNA amounts per mass unit. An alternative method that has been considered is to express the results as number of copies of the insert as a percentage of the number of copies of an endogenous reference gene. But here, the problem is that the results will be dependent on the ploidy level of the plant. The European Union now recommends that quantitative PCR results are expressed in terms of DNA copy number per haploid genome. That can work where PCR is used as the detection method, but it has to be recognized that this will give a different answer from protein-based analysis, which can only be expressed on a weight-to-weight basis, i.e. the amount of the GM protein as a percentage of the total protein. The methodologies are still evolving, but this debate has to be placed in the context of the fact that the threshold for labelling is in any case a somewhat arbitrary decision and varies between countries. So I end with a few final thoughts. As I've described, sampling and testing present many difficulties, and results will affect the validity of any labels. Labelling incurs costs, which will be passed on to the consumer in terms of higher food prices. Countries need to take decisions on whether and why labelling is required. They need to decide on voluntary or mandatory labelling, on whether the may contain or does contain option is to be implemented. In any case, labelling of foods sold in the informal sector is impractical, and if regulations are not implemented in the informal sector, is it appropriate to require labels for the formal sector? And finally, any labels need to be appropriate for the audience in terms of language and content, not to raise fear and alarm. In this final slide, I give links to a few references that may be of interest. Obviously, this list is by no means exhaustive, but it can be a useful start for those of you who wish to explore the subject further. Thank you.